Hello, everyone. My name is Matvey Bulat. On behalf of the CNBR Open team, I'm glad to welcome you to the next seminar devoted to the brain's novelty and routinized perception. While today's participants connect to our broadcast, let me say a couple of words about CNBR Open for those who participate in our seminars for the first time. Well, the CNBR Open project unites people interested in or engaged in neuroscience and neurotechnology. And the name may seem a little strange, but CNBR actually stands for the Center for Neurobiology and Brain Restoration of the Skolkovo Institute of Science and Technologies. Well, this is the young and modern research center in Moscow. And this is where the project was born and is working for more than a year. The CNBR Open project's goals are to allow first students and enthusiasts to learn and interact with professionals, professors, and scientists working in the field, look after a laboratory, establish contact, learn from them, etc. And the second goal is to connect business and academia. Participants in our projects, they are representatives of international companies on the one hand, and experts and project curators on the other hands. And on our side, they find each other, which sometimes translates into different sorts of mutually beneficial outcomes. And third goal is rallying into rallying neuro community, in which it's interesting to communicate on professional topics, to come up with and effectively implement projects in collaboration. In this sense, we actively collaborate as a seminar with the international community, including the Neurotech X organization. Well, that's almost it. So we have attendees still coming, but let's begin. Well, if you want to know more, just subscribe to the project news and participate in our future events. Besides open seminars, we also meet in online and offline neurobars in different cities, at the conferences, events and presentations, etc. Before we start, I would also like to notice that we have quite a lot of participants, so we cannot do this seminar in Zoom meeting. Instead of that, we put everything into Zoom webinar. If you have any question, please put your question to Q&A on the bottom of your screen, not to the chat. And so today's speaker will answer all the questions after his talk. At this time point, let me switch myself off and invite to today's floor and stage our special guest for today. Well, we have a speaker from New York today and a special guest, Professor Vladimir Zelman from California, whom I would like to ask to say a few words. So, as a professor and the founder of CNBR, and Professor Zelman, could you please turn on your video?
Yes. No, for everybody is probably a good evening or before evening. For me, it's early morning, and it's a special privilege today to tell a couple of words about our center. It was created a couple of years ago, I believe. The idea came exactly 8 or 9 years ago when the Skolkovo Foundation was organized and the Institute of Science and Technology, Skolkovo Institute of Science and Technology. But very interesting that a couple of weeks ago, I received an email from Professor Goldberg from New York University, who asked me what kind of center is this and how relationship you are. And I told, oh, yes, this is an interesting center. We just organized, it's a young one. And we try to develop a new center in Moscow, which is famous for neuroscience. Actually, the first Nobel Prize laureate was Ivan Petrovich Pavlov from St. Petersburg. Anyway, and then he started to speak with me a little in Russian and told, I believe heard about you. You know Alexander Ivanovich Luria? And I told, of course I know, because some more than 50 years ago, he came to us with ideas to study the brain to the Institute of Neurosurgery, where Professor Sebenenko introduced at that time a very unique, interesting method how to measure the cerebral blood flow. And the most probably famous scientist in neuropsychology was, I believe, in that time and still is the father of neuropsychology, is Alexander Romanovich Luria, which I know him personally and I remember very well. Anyway, by this Dr. Elkhonon Goldberg, who actually organized Institute of Luria Neuroscience Institute in New York. And we start to remember things, and I believe it was more than 50 years ago. We even met somewhere in one of the laboratories in the neurosurgical Burdenko Institute, which is probably one of the biggest in the world right now. And this is how we became connected in discussing our cooperation. Actually, Dr. Goldberg already was in touch with the associate director of the Institute of Medicine and Psychology in Novosibirsk University. And I believe we can work out a very interesting together international program. I would like right now that Professor Lebedev, Mikhail Albertovich, should introduce our guest speaker and present him his contribution to neuroscience. And I again thank you very much, Bulat, that you invited me, Matvey. And I hope that we will have a long, very productive cooperation.
Thank you, Vladimir Lazarevich, for joining us today.
Hello, it is my pleasure and great honor to introduce today's speaker, Elkhonon Goldberg. He's a famous neuropsychologist and cognitive neuroscientist, and he is very famous for his work on hemispheric specialization, and particularly on the novelty-routinization theory, which he will be talking about today. So Elkhonon studied at Moscow State University, and as Vladimir already mentioned, he worked with Alexander Luria. And at that time, Elkhonon work was in the field that we call now computational neuroscience. So he can be considered a pioneer of computational neuroscience. He moved to the United States in 1974. And from that time, he worked mostly in clinical neuroscience. So he studied the functions of frontal lobes, hemispheric specialization, memory, cognitive aging, and theory of functional cortical organization. He is currently a clinical professor of neurology at New York University School of Medicine. And he is also a co-founder and chief scientific advisor of Sharp Brains. This is an online brain fitness center. So the idea here is that brain plasticity can be used to delay or even remove the effects of cognitive aging. Elkhonon is also a founding director of Luria Neuroscience Institute. This is an organization with the purpose of advancing research and dissemination knowledge about the brain and the mind. According to Wikipedia, he is an atheist with agnostic tendencies. Elkhonon authored 6 books. I will probably name the titles. So it is this, the first book is Contemporary Neuropsychology and Legacy of Luria. Then the second book is The Executive Brain, Frontal Lobes and the Civilized Mind. And then the third book is The Wisdom Paradox, How Your Mind Can Grow Stronger as Your Brain Grows Older. Then The New Executive Brain, Frontal Lobes in a Complex World. And the book that was published just recently is The Human Brain in the Age of Innovation. So you can see that today's speaker has a huge contribution to neuroscience. So uh, let's listen to the talk.
Hello, good afternoon, good morning, wherever you are. It's an honor to participate in this event. It's a nostalgic event for me. This is the first time I am addressing a Russian audience in many years. And so appropriately so, the story that I'm going to tell started in Russia many years ago when I was still a student at University of Moscow, almost half a century ago. And it remained an enduring theme of my work up to this day, all right? And the story started when I was probably 19 or the years of age or thereabouts, when I was told what all of us are told, that the brain consists of two hemispheres. Now, the question is, how come I cannot move my slides? Okay, now I can. When I was told what everybody is told, that the brain consists of two hemispheres, that the left hemisphere, the dominant hemisphere, is in charge of language, and the right hemisphere is subdominant hemisphere, and it's in charge of various non-verbal functions. And from the moment I heard this, I was skeptical. I didn't feel that I had any reasons to believe that this the formulation was wrong, but I was quite convinced from the get-go that it had to be incomplete, okay? And the reason was that, you know, a little that I know about the neuroscience at the time, and I was only beginning to learn, I knew that that the brain consists of two halves across all the species, that it's not a unique human feature, that it's a ubiquitous, universal feature of brain architecture across multiple species. And then I thought, if this bicameral, hemispheric architecture has been conserved across evolution as a structural feature, then there had to be also a functional attribute of these two hemispheres, which would also be conserved across evolution. And on the other hand, this distinction between language and nonverbal functions is devoid of any meaning outside of our species. It's only meaningful for humans because other species don't have language. So I thought that even though this distinction between language and nonverbal functions probably captures some properties of the human brain, there has to be a more general principle of which this language-non-language dichotomy would merely be a special case, which would be applicable throughout evolution across species. And I have no idea what this more general principle was, but I was sure that there had to be one, okay? And so, and indeed, much later, we learned that the two hemispheres not only exist in the brain as kind of a major architectural principle, but that they are also quite asymmetric. They are asymmetric. Here on this slide, I'll list a bunch of asymmetries which characterize the two hemispheres. And the important point that I want to make is that none of them is unique to humans, that we share each of these asymmetries with multiple mammalian species, other species, okay? What are these asymmetries? So-called Yakovlevian torque, whereby the right frontal lobe is a little bigger and sort of protrudes over the left frontal pole, and the occipital pole is slightly bigger and protrudes over the right occipital pole. Planum temporale, which is roughly speaking, which is larger in the left hemisphere. The frontal operculum, which is also larger in the left hemisphere. So there's a whole bunch of these morphological asymmetries, which we share with other primates, at least, and possibly even with other simians, okay? There are also, ahead of my slides, there are also cellular differences, asymmetries, so-called spindle cells, for instance, phonoconomo cells, which are very interesting cells. They have large bodies. They are located only in the frontal lobes. They are located throughout the prefrontal cortex. Initially, people thought that they are only found in the anterior cingulate cortex. We now know that they also extend, they are found also in the prefrontal convexity and orbitofrontal regions, but their distribution is limited to the prefrontal cortex. They are found only in the great apes, including humans, and in dolphins and in elephants, in creatures with very large brains, very complex cognition, very complex social cognition. And these cells, these unique cells, are distributed asymmetrically. They are much more prolific in the right hemisphere than in the left hemisphere. Again, appears to be an enduring invariant feature across these multiple species. So in addition to morphological asymmetries, there are also cellular asymmetries. There are biochemical asymmetries. Major catecholamines, norepinephrine appears to be more prolific in the right hemisphere, and dopamine appears to be more prolific in the left hemisphere. Again, a feature which we share with lowly creatures like rodents, for instance, okay, not unique to humans. And more recently, we are beginning to find out, and we have some papers on the subject, that there are also the various genes are expressed in the brain asymmetrically, okay? So you have this whole collection of attributes which are characterize the two hemispheres asymmetrically and which we share with many other species. And so just to complete the picture, this is from our own work. I don't want to dwell on these slides. Time permitting, we'll talk more about them. At this point, I just want to show them these slides. And these are morphometric studies conducted using magnetic resonance imaging in humans. And we were interested in relative sizes of various structures in the two hemispheres. And here, just this is a simplified version of the slide. Aquamarine areas are larger in the left hemisphere. Yellow areas are larger in the right hemisphere. These are males, these are females, which is an interesting aside for which we don't have the time today, gender differences or sex differences. This is humanity as a whole. And you see that a number of regions exhibit a kind of an asymmetric size, okay? So with that in mind, I thought, since there are so many attributes of the two hemispheres which are asymmetric, and these asymmetries are conserved across various species, there has to be also a parallel, as I said, functional asymmetry. And this functional asymmetry cannot be captured simply by this distinction between language and nonverbal functions. The simple reason being that this distinction is devoid of any meaning outside of our species. And there had to be some fundamental distinction which would be meaningful across these various species. But I had no idea what this kind of a hidden general principle was. And so all these ideas were percolating in my 19-year-old head. And then a few years later, I began to work, as Professor Zelman mentioned, at Burdenko Institute of Neurosurgery, in the lab of my mentor and teacher, Alexander Luria, who was a professor at the University of Moscow, where I was a student, but his lab, our lab, was at the Burdenko Institute, where we were learning how to examine patients and conducted various research. And there I struck friendship with a few pediatric neurosurgeons, young guys also, maybe a little older. I was in my 20s, they were in their 30s. And one day, one of them told me, he said, listen, we are beginning to encounter something which runs counter to everything we were taught in medical school, okay? We were taught in medical school that the left hemisphere is the dominant hemisphere and the right hemisphere is kind of a less important hemisphere. But what we're finding in our clinical experience goes counter to that, okay? We're finding that at least in children, damage to the right hemisphere is much more catastrophic than damage to the left hemisphere. And of course, everybody was taught that at least in adults, it's the other way around. The damage to the left hemisphere due to tumor, head trauma, or stroke, or whatever, is usually much more catastrophic. So there was this interesting contrast between the effects of brain damage, lateralized brain damage in adults, where damage to the left hemisphere was more catastrophic, and everybody knew that. And in children, as these neurosurgeon friends were telling me, where damage to the right hemisphere is much more catastrophic. So they said, well, so you are a neuropsychologist, you sort it out, you figure it out, and let us know, okay? And so And that kind of a provided fodder for kind of a mental gymnastics. So I began to think about it and came up with this idea, which turned out to be a very productive idea, and it sort of guided some of my work through for the duration of my career up to this day. Namely, that perhaps it is the case that there is a kind of a fundamental functional distinction between the two hemispheres, such that the right hemisphere deals with cognitive novelty, and the left hemisphere serves as the repository of cognitive routines. And of course, in infants and very young children, the world is completely novel. The collection of cognitive routine is very limited and basically zero. And then as one develops and grows and accumulates information and knowledge and skills, the domain of novelty shrinks, and the kind of repertoire, the arsenal of cognitive routines increases, and as a result of that, there is this kind of a gradual shift of the kind of a locus of cognitive control, if you will, from the right hemisphere to the left hemisphere. So that was the idea which sort of captivated me as a hypothesis, and indeed, what these young neurosurgeons were telling me in Moscow many years ago, that was their eyeballing. It was based on their informal observations. It was not a product of some kind of formal research. But years later, it turned out that they were right, that some formal studies have coming out mostly out of the United Kingdom, basically confirmed what they were sensing, what they were seeing in their patients, that really in little children, damage to the right hemisphere is more catastrophic and more kind of consequential in its negative consequences than damage to the left hemisphere. These were studies conducted with early hemispherectomies with children who, because of intractable seizures, had to undergo the radical neurosurgery, whereby a whole hemisphere was taken out. And at that time, I made the decision to move to the United States. I moved to the States in 1974 with the plan to basically launch this research program on this hemispheric specialization, guided by this distinction between cognitive novelty and cognitive routinization. And of course, we did with a colleague, Louis Costa, much older. He was a dean at a university at the time, and I was just basically a guy in my late 20s with 2 briefcases and $200 to my soul, who just arrived on the American shores. We decided before embarking on, and I sort of infected him with this idea. So we decided before embarking on an empirical work, on research, to conduct literature review, and basically we concluded that everything that we set out to actually demonstrate experimentally already existed in the literature. In bits and pieces, it was a matter of finding it and sort of putting it together in a kind of coherent context, which we did, which resulted in this paper, which we published many years ago, titled Hemisphere Differences in the Acquisition and Use of Descriptive Systems, where we basically articulated this idea. And the idea basically is kind of captured on this slide. We posited that the right hemisphere is critical for exploratory processing of novel cognitive situations to which none of the previously formed representations or cognitive strategies is readily applicable. And the left hemisphere is critical for processing with reliance on pre-existing, well-routinized cognitive strategies and representations. And this is kind of a diagrammatic, conceptual representation of this whole idea, so that when an individual encounters a totally novel situation, novel cognitive challenge, the right hemisphere is the dominant one, and then the individual develops some cognitive routines which are task appropriate to that class of tasks. Then the left hemisphere takes over, and these processes we posited occurred on multiple time scales for various processes. It's not a single curve, but it's a multiple curves kind of unfolding on multiple time scales. And of course, the ramifications of this formulation are multiple. First of all, it implies a greater degree of individual differences, okay? Because how are cognitive neuroscientists to this day even, and neuropsychology certainly half a century ago when I went to school, were taught. We were taught a litany of cognitive skills which resides in the right hemisphere, and another litany of cognitive skills which reside in the left hemisphere, and that was supposed to be static and inviolate, okay? But we were saying something else, that in reality, what may be mediated by mostly by one hemisphere in Johnny may be mediated by the left hemisphere in Tommy as a function of the relative degree of familiarity with these cognitive situations. So much greater degree of individual differences, much greater degree of intrapersonal differences over time. In other words, what today may be sitting mostly in my right hemisphere in due time will somehow migrate to my left hemisphere as I develop some appropriate cognitive skills. Less absolute functional localization. And to this day, you see, in my kind of a professional life, I wear two hats. I try to be a cognitive neuroscientist, but I'm also a clinician, kind of a doctor who sees patients, okay, with various forms of brain damage. And to this day, I'm very skeptical about very, very kind of a precise localization. When I read in various research papers, and today we see a proliferation of these research papers with the advent of functional MRI, where claims are made about very, very specific localization of various cognitive functions, okay? Whenever I see that, I basically refuse to believe it. I think that it's bull. I think that it's, and this skepticism, I was infected with this skepticism precisely because of this kind of the kind of a theoretical position that we developed and now. So less absolute functional localization. And what was particularly attractive to me, at least in this idea, is basically that which served as the impetus behind this idea, namely that this distinction between novelty, cognitive novelty and cognitive routines is meaningful across multiple species. Unlike this distinction between language and nonverbal functions, which completely are devoid of any meaning outside of our own species, this distinction between cognitive novelty and cognitive routines is meaningful for any species capable of learning, okay? And this is what made this, and then you can very readily understand this functional distinction between language and nonverbal function as a special case of that much special case applicable to humans, but not outside of our species, which is just a special case of this more general, more fundamental principle, okay? So, so now the story that I'm going to tell you is the story of two parts. It's a story of neurobiology of novelty and neurobiology of cognitive routines. So let's proceed, neurobiology of novelty, all right? So basically what we posited as a kind of a far-fetched hypothesis was gradually finding support. Some of this support came from neuroimaging. So this is a study, for instance, by a French scientist, Chiron, who used single photon emission computerized tomography. And what I'll be doing, I'll be traveling in time. You know, this whole kind of a little saga of mine started in the late 60s when I was a student at University of Moscow. We continued this work over the years and decades. And so you will see that some of the research, both our own and research by other people, is old, some is very recent, and some is somewhere in between, okay? Because it's an enduring kind of a theme which unfolded as a coherent narrative, right? So a study by Chiron using SPECT, single photon emission computerized tomography, where she demonstrated that in very little children, the right hemisphere is physiologically more active. And then, of course, SPECT tells you about blood flow. And then, of course, which people regard as a kind of a useful proxy measure of neural activity. And then at the child age, after the age of 3, there is this gradual shift of kind of the center of physiological activity from the preponderance of the right hemisphere to the preponderance of left hemisphere. Exactly what we would have predicted, okay? The neurotransmitters. These two major neurotransmitters, catecholamines, norepinephrine and dopamine, as I said earlier, they somewhat lateralize. Norepinephrine is slightly more prolific in the right hemisphere. And of course, we know, and multiple pharmacological studies pharmacological challenges have demonstrated in various animal models that when you stimulate the noradrenergic system, the animal engages in orienting behavior, in exploratory behavior. Okay? With dopamine, it's the other way around. Dopamine is more prolific in the left hemisphere, which leads to all kinds of interesting ramifications for diseases such as Parkinson's disease, Tourette's, but we will not talk about that. We don't have time to talk about it now. And when you stimulate the dopaminergic system with pharmacological challenges, you create stereotypic behaviors. The animal begins to run in circles. You stimulate norepinephrine, the animal explores. You stimulate dopamine, the animal engages in stereotypy. The opposite behaviors and opposite the patterns of hemispheric asymmetries. Neural net modeling, as Professor Lebedev mentioned, I started my career, attempted to start my career in what today is called computational neuroscience. In those days, this term did not exist. Now, I became very captivated by neural nets, by this classic work by McCulloch and Pitts, and I decided that that's what I wanted to do for a living, and created a special program for myself, whereby I was a student in the psychology department at MGU at University of Moscow, but also I had a special program in the Mehmat department of mathematics, so I studied both. But it was sort of premature. I mean, there was no context. But I've retained this interest in neural nets. Now it's vicarious interest. I'm hopelessly behind the curve, but conceptually I try to follow the field. And so a good friend of mine, Stephen Grossberg, who is one of the kind of pioneers of neural net modeling in the United States, posited that in order to create a computationally efficient neural net, you need to separate these two aspects thereof. That aspect of the neural net which is in charge of acquiring new information, and that which is in charge of storing previously formed information. Because evidently in these computational devices, there is certain tension, as it's very difficult to acquire new information without degrading previously acquired information. This is a big kind of a quandary for all these neural net designers. And so both Grossberg and more recently these people from University of Colorado in Boulder came up with this idea that in order to resolve this tension within the neural net, you need to actually spatially separate these two aspects of processing, storing previously acquired information as opposed to acquiring new information. Otherwise they clash, okay? And so it's very likely, I thought, that the evolution solved this dilemma by sort of investing the two hemispheres with these two different aspects of information acquisition and storage, because the brain is a neural net. That's where, yeah. So it was a very attractive, at least to me, thought that perhaps perchance evolution solved this dilemma by allocating these two functions, storing information versus acquiring new information, to different hemispheres. Yeah. And finally, experimental work, both some very old-fashioned experimental work. But as I said, this whole saga started in the late 60s, so it's not surprising that I'll be talking a little bit about some very old work. Basically, we were all taught that certain functions are mediated by the right hemisphere, facial recognition. Everybody, every neurologist, every neuropsychologist taught that when there is damage to the right hemisphere, prosopagnosia developed, a syndrome of whereby an individual loses the ability to recognize faces. Amnesia, ability to recognize music, has also been linked to the right hemisphere, and damage to the right hemisphere ostensibly causes that. Yes, but as experiments have shown, it's true and it's not, okay? That it's true for, with respect to amnesia, it's true for kind of the majority of humanity. Most people are not trained musicians. And indeed, if a lay individual suffers damage to the right hemisphere, that's when the ability to process music will suffer. In trained musicians, it's the other way around. damage to the left hemisphere will result in impairment of musical processing, and damage to the right hemisphere will not. Something similar has been described in with facial recognition. It's true that damage to the right hemisphere will result in your inability or your impaired ability to recognize new faces. You walk down the street, you bumped into some Joe Blow, then you bump into him 10 minutes later, and you recognize him as the Joe Blow who stepped on your foot 10 minutes earlier, okay? This kind of facial recognition will be impaired following damage to the right hemisphere. In contrast, recognition of faces, famous faces, faces that stare from you for magazine covers, have been staring at you for magazine covers for years and decades, will be impaired following damage to the left hemisphere. John Kennedy, Winston Churchill, Joseph Stalin, whatever. Famous individuals who have been staring at you from magazine covers and pages of textbooks are processed differently, are processed with a particular reliance on the left hemisphere. So the same idea, the same principle that novel faces and novel music or these processes for individuals who are not trained, who don't have some special skills dedicated to these cognitive domains, are mediated by the right hemisphere, but in individuals who, with extensive training in a certain cognitive domain or extensive exposure to certain classes of stimuli, similar information would be mediated by the left hemisphere. Again, goes again this kind of a static notion of kind of an absolute invalid link between certain functions in certain hemispheres. Okay. Work conducted to a large extent by my friend and actually former student, he was my graduate student, Alex Martin at NIH, and some other people using PET. I don't want to go into this great detail, but basically what they demonstrated was positive emission tomography. Basically what you can do with various neuroimaging devices, functional neuroimaging devices, fMRI, PET, SPECT, you name it, EEG, okay? You can place a subject into this experimental setup. You can introduce various cognitive tasks to the subjects. The subject is doing these tasks, and meanwhile you're recording that individual's cognitive activity, neural activity. And you can basically, this technology allows you to monitor, to let your subject engage in this activity for up to maybe 40 minutes. And so in the course of you can construct your experiment in such a way that it starts as a totally novel enterprise. And then in the course of this half an hour, 40 minutes, there is certain familiarization, and you can examine various changes which may occur in the brain. And so there have been a number of such experiments, and they using various tasks. And I don't want to go into, and they all demonstrated that with the task familiarization, there was this gradual shift of the preponderance of neural activity from the right hemisphere to the left hemisphere. It turned out to be the case for memory tasks. So there was this shift involving the hippocampi, for visual tasks involving occipital cortex, right to left occipital, for various tasks of working memory involving the prefrontal cortex, etc., etc., etc. So it used to be, it was really demonstrated that it's a highly universal phenomenon, okay, not linked to any particular task. In normal development, it turned out that yes, in adult individual languages lateralized to the left hemisphere, but in little children, it's not less lateralized. And there is even some evidence that certain aspects of language are mediated mostly by the right hemisphere in very little children. And in lesion effects, I already showed you this slide, it appears that damage to the right hemisphere is much more catastrophic, confirmed the hunch of these neurosurgeon friends of mine at Burdenko many years prior. Damage to the right hemisphere at a very early age is more catastrophic to subsequent cognitive development than damage to the left hemisphere. Okay, let's skip this. So generally, it turns out that what is and there were some also interesting studies by Aaron Smith at University of Michigan, who studied lifelong these individuals who lost one or the other hemisphere very early in age. Suppose somebody developed infantile seizures. In those cases, sometimes the decision is made to engage in very radical surgery to take out the whole hemisphere. And that means that the whole individual development occurs subsequently with the reliance on a single hemisphere. And it turned out that an isolated right hemisphere results in a higher IQ than the isolated left hemisphere. Okay, so the question is, this whole notion that the left hemisphere, the dominant hemisphere, is quite misguided. And finally, our own work conducted using fMRI, functional magnetic resonance, in collaboration with Kai Vogelei, a colleague of ours from Germany, basically demonstrating that when the task is to normal subjects to observe various stimuli and identify similarity, in other words, when your attention is drawn toward looking for the familiar, you have an activation mostly in the left hemisphere, these green blobs. And when using the same stimuli, the task is to pay attention to differences, to kind of a novelty. What you have are these blue blobs. The activation is in the right hemisphere. A study conducted in Japan in collaboration with us using EEG. They looked at gamma frequency, and that 30 to 40 hertz, very high frequency, which is typically associated with mental effort. You have a kind of an increase in the power range of this frequency when an individual is engaged in some strenuous cognitive activity. And this is kind of a natural history, Brutus, you have to stop. I apologize. I have my dog here, and he began to slurp. He began to drink his water. Yeah, I hope you didn't hear it. So an individual is engaged in a novel cognitive task. We developed a whole collection of tasks, which were called agent-centered tasks. I'll explain them a little bit later. It starts as a novel cognitive task, and then there is a familiarization with the task. And basically, in the course of this experiment, in the beginning, when the task is novel, you have an activation in the prefrontal cortex and in the right hemisphere, and then it becomes increasingly familiar. The center of cognitive gravity, if you will, shifts away. So it's a kind of a story of two parts. It's a bidimensional shift. And today we're talking about only one and kind of ignore the other one. There is kind of the center of cognitive gravity moves away from the prefrontal cortex to the posterior cortex. This is not part of our story today. It's interesting in its own right, maybe kind of a topic for another time. And also what is of interest to us here is that there is the shift of cognitive, kind of a center of cognitive gravity from the right hemisphere to the left hemisphere, okay? So with the, now this is, I think, is a very kind of a dramatic illustration of how with the increased familiarization, there is a kind of a reduction of involvement of the right hemisphere and the increase of involvement of the left hemisphere, right? It's kind of, you see, it's kind of, a picture is worth 1,000 words. So here you have this picture, yeah? And another study conducted by my former student, Vasilisa Skvartsova, a Russian girl, well, no longer a girl, she is now, I actually lost track of her. She was at NYU. I think she's now at University College in London. Where it was an fMRI study using Wisconsin card sorting test. If there are perchance neuropsychologists among them, it's one of the most commonly used cognitive tasks, okay? Basically, it's a guessing game. You need to kind of uncover certain hidden principles, and it is sort of a fractal task. First you have to kind of figure out that the underlying principle changes, kind of a macro solution, but then you have to discover a specific principle 6 times. If you figure out this basic principle, that this underlying principle is not static, that it changes over time, then you have done half the task, but still you have to solve these specific tasks. So it was used as an activation task in fMRI, and what she did, basically she wanted to in functional resonance, in fMRI work, there is a very specific kind of or in any functional neuroimaging work, there is a very kind of a specific type of challenges which arises. You need to separate this activity, that activity which is unique, specific to your task, from some extraneous activity. Because if your subject lies in the scanner and is doing this task, there may be all kinds of other things happening in the subject's task. He may be thinking about, he may be having a toothache, he may be thinking about his failing marriage, God knows what, okay? So how do you know which aspects of this activity are related to the task and which are extraneous? So a very elaborate methodology has developed, to which people sometimes refer to as subtraction methodology, and which may take many forms. Has gone much beyond simple subtraction over the last, I don't know, 10, 15 years. But basically what she did, and she did it twice. So as I said, the subject needs to develop, to identify, to uncover 6 categories. So what she did, she took the first 3 categories, averaged across them, then separately took the last 3 categories, averaged across them, and subtracted the second average from the first one. By so doing, she identified those parts of the brain which were, and when I'm saying subtraction, it's really comparison, voxel by voxel. You look at kind of compare activation levels in different voxels, okay? And by so doing, she identified those parts of the brain which were active in the beginning, but stopped being active toward the end of the sequence. And guess what she found? Well, this is a radiological convention, the common one, left is right and right is left. She found activation in the right prefrontal regions, okay? As I said, prefrontal is for another day, it's a separate story, but what I want to emphasize now is that the activation was in the right hemisphere. And then she did something similar for each category. For each of these 6 categories, she split each of those into the first half and second half, averaged across 6 first halves, averaged across 6 second halves, subtracted the latter from the former, and basically, and by so doing, identify those parts of the brain which were particularly active at the beginning of the discovery of each of these specific categories, and basically found the same thing, okay? That the activation was in the somewhere in the right hemisphere, so right prefrontal regions. So it goes to show that in this task also, it appears that when the task is novel, it is the right hemisphere which is particularly active and not the left hemisphere, okay? And then with task familiarization toward the end of the task, where there is certain familiarization, this right hemispheric activation dissipates. Another line of work was conducted by my good friend Alan Snyder, who is a transplanted New Yorker who found himself in Australia. And we became very close friends. I used to be a visiting professor at University of Sydney for a number of years, so I used to spend a lot of time in Australia. So basically, he used various techniques to manipulate the brain, neurostimulation used such as RTMS, repetitive transcranial magnetic stimulation, and transcranial direct current stimulation. These relatively commonly used techniques to modulate the brain. Basically, he used it to suppress the left hemisphere. Basically, at least now this methodology evolved very rapidly over the last few years. These studies were done probably 7 or 8 years ago or thereabout. Generally, with transcranial magnetic stimulation, the idea is that with low frequencies, maybe 1-2 hertz, you suppress the underlying tissue. With high frequencies, like maybe 10-20 hertz, you activate the underlying neural tissues, okay? So, and for therapeutic purposes, like to treat depression, people use high frequencies, okay? But in his experiments, he used low frequencies, basically creating transient physiological lesions. And so he did that to basically transiently, temporarily wipe out the left hemisphere, the left temporal parietal regions, claiming that ostensibly somehow disinhibiting the right hemisphere, because of course the corpus callosum contains both in excitatory and inhibitory pathways, but there are more inhibitory pathways. I forget the exact ratio. I think it's 6-4. Okay, 60% of the pathways. I don't hold it against me if I'm wrong, but the majority of pathways are inhibitory. So by wiping out one hemisphere, you, in some sense, stimulate the opposite hemisphere. Okay, for the duration of this manipulation. And what he claimed, and these are published data, I produced it in my book, but basically it's from his published journal articles, that by so doing, he enhanced the subject's ability to solve various out-of-the-box tasks which require some kind of a very original kind of a turn of thought, which are usually quite intractable to most people. Okay, how to combine these 9 dots with minimum number of lines. Okay, usually people kind of even though it's not part of the instruction, they tend to stay within the perimeter of this rectangle, even though nobody tells them that they have to. And the moment you kind of go outside of this perimeter of this rectangle, you can connect these dots with only 4 lines. And evidently, kind of a normal human people who are not some kind of quasi geniuses find these tasks very difficult. That's out of the kind of it's a well-known task used in cognitive psychology to study out-of-the-box thinking. So by, according to Allen, by suppressing the left hemisphere, he somehow enhanced the subject's ability to do that. And similar task, this mental matchbox arithmetic, when you have to kind of turn one equation into another by just moving a single matchstick. And evidently people find these tasks also very difficult. And with the when he impacted the left hemisphere, negatively impacted, suppressed it, he had this ability. Now, and I talk about it in this book of mine, which came out in the United States 2 years ago in 2018, and came out in Russia. Kreativny mozg, this is hardcover and paperback, also Kreativny mozg, published by Exmo. Okay, now, and at this point, I need some guidance how we're doing on time. My talk is a modular talk. So if we need to quit, this is a natural point in time when we can quit, because my next little kind of a module was the neurobiology of cognitive routines. If you recall, it's a, the talk is of 2 parts, neurobiology of novelty and neurobiology of routines. We can proceed with the routines, or we can proceed with the questions. And somebody probably Matvey should give me some guidance. If I don't hear from him, I'll just Matvey.
Yeah, we have several questions already, so we can maybe do the following. Let's answer these questions and then go to the second part.
Okay. So you direct traffic. Mm-hmm.
Okay, so if you click on the Q&A on the bottom of your screen, you will also be able to see the questions.
Hang on. Yeah. Mm-hmm. Yeah. Okay. Hello. Shall I read them out loud? Pedro Ferreira. Hello. How can I get the recording after the presentation? That's a Matvey question.
Yeah. So I will answer it in a type form. Let's proceed.
Yeah, you certainly have my permission to record and to distribute this stuff. Yeah. Grigory Petrov, Attention Schema Theory by Michael Graziano evaluates attention dynamics as they learned by our brains. Also pinpoint as one of the possible neuronal correlates of cognition. Have you checked this? Well, yes, I am somewhat familiar with this work. Well, neural correlate of cognition. Cognition is like an ancient deity. It's everywhere in the brain. Okay, saying that it's somehow linked to a particular region would be an oversimplification. However, I believe, and I agree, and in fact talk about it in this book of mine on creativity, that a solution of novel problems does critically involve the right hemisphere and involves temporoparietal regions in the right hemisphere. Okay. Are other neurotransmitters besides norepinephrine, such as serotonergic neurons, are also asymmetric in the hemispheres? Not to my knowledge. It's entirely possible that serotonin also is, but not to my knowledge. Um, right, anonymous. Sir, can damage induce positive facts, like a person who was not interested in music, but after injury, he grows interest in music? Matvey would like to answer this question.
I have actually no idea, but I guess that it can be possible.
It says Matvey Bulat would like to answer this question.
Ah, no, no,
Somebody
no.
who is
This is just I'm clicking this so that the other
I
participants
see.
can
Well,
take a look.
okay, bizarrely so. Bizarrely so. The answer is yes. There's been some interesting work, and I talk about in my creativity book also, pertaining to a particular form of dementia, frontotemporal dementia. Frontotemporal dementia is a very interesting type of dementia, uh, which is characterized by an earlier onset than, let's say, Alzheimer's disease or Lewy body disease. It affects the frontal lobes and or let me be more specific. It affects orbitofrontal cortex and or anterior aspect of the temporal lobe. That's why it's called frontotemporal dementia. It tends to um, uh, uh, affect the left hemisphere more than the right hemisphere. I'm sure there are plenty of exceptions, but if you average across many patients, you will conclude, in fact, we have a whole paper on the subject, you will see that the left hemisphere is more vulnerable. And there have been some anecdotal cases reported. There is a very kind of a interesting group. Bruce Miller is a neurologist at UCSF, University of California, San Francisco. His group and some other people reported some anecdotal cases of individuals who developed frontotemporal dementia and exhibited some, all of a sudden exhibited some artistic talent which they had not been known for before, okay? Like musical talent or literary talent or painting. But of course, that was a fleeting phenomenon, and then they succumb to dementia. So this is a very heavy price to pay for this kind of a moment of 10-minute moment of artistic talent. But the answer is yes, in this kind of a bizarre kind of way. Yeah, AC. During
Dr.
right
Goldberg, I propose to take one more question and then proceed with the rest of the lecture, and we will return to the questions in the second part of
okay,
the talk.
okay. During right to left hemisphere perceptual transfer, connectivity, directional asymmetry. Uh, I'm not sure I understand the question. Well, it's actually, to the extent that I do, it's a very interesting one. I am not aware of any ability to do that, but maybe there may be some technology that would enable you to see basically in which direction activity moves in the core. Right. Yeah, the answer is probably yes. I'm not aware of this study, but actually it would be a very interesting study that you came up with, Mr. or Miss AC, to do some recordings from the corpus callosum to try to see the direction of action potential. Because there are pathways going from left to right and pathways from right to left to see in which direction there is more activity. I'm not aware of such study, but it would be very interesting. Maybe you should do it. Okay, now Matvey, we continue with this lecture.
Yeah, let's continue. And I
How
would
much
also
more?
like to say to the participant that in the second part, we will give the opportunity to raise a hand and ask a question orally as well. So I see several professors like Alexander Kaplan and Yuri Kotelevtsev, for example, in the participant list. So everyone will be able to also raise a hand, not to type.
How much time do I have, Matvey, so that I know how to submit?
Yeah, half an hour approximately.
Okay, very good. Okay, so let's move on. So I switch this off, yeah, like this. Okay, so neurobiology of cognitive routines. Okay, somebody needs to let me move my slides. Matvey,
I'm not stopping.
you're not stopping, something is. Okay, now they're unstuck. All right, so we'll continue this train of thought, and just as we kind of posited that the right hemisphere is the medium of dealing best equipped to deal with cognitive novelty, uh, 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 we will now, I'll present some evidence that the left hemisphere is the repository of established knowledge and skills, okay? And It started with a clinical case. Many years ago, how many years ago? Sometime in the very late 70s, maybe 79 or thereabout, I get a call from a friend of mine, Jim Hughes, he's since passed on, a neurosurgeon in New York, who wanted me to see a patient of his. And as I said, I try to be a scientist for better or worse, but I also do clinical work. I see patients. A man in his mid-30s who was in a horse riding accident here in New York in Central Park suffered significant brain damage. And as we began to examine this patient, we concluded that he represented a neurological impossibility, okay? Because according to every neurological canon, according to every neurology text or neuropsychology text, retrograde amnesia cannot exist in isolation. Of course, for those who don't know this, we distinguish between two kinds of memory impairment. We talk about anterograde amnesia, that's memory impairment. It's characterized by an inability to learn new information following the onset of brain damage, as opposed to retrograde amnesia, an impaired ability to access information presumably acquired before the onset of brain damage, okay? So according to every neurological canon, that's what I had been taught in my courses at University of Moscow, and that's what every neurology text said. Anterograde amnesia is invariably more severe and more enduring than retrograde amnesia. Anterograde amnesia can exist without retrograde amnesia, but isolated retrograde amnesia without anterograde amnesia is not supposed to exist. Well, guess what? In this guy, it existed, okay? It was an unusual case so that you would approach him. In the beginning, he had very profound anterograde amnesia. I would approach him, introduce myself, then he would forget my existence, ever having met me maybe half an hour later. And he had profound retrograde amnesia. He thought that he was 17 years of age. He would give you a very lucid kind of his life story up to that age. He would ask him, where do you live? He would give his parents' address. What do you do? I go to high school. In reality, he went to an elite university, and he had a very impressive corporate career. He had a wife and several children. He was totally incognizant of any of that, okay? Well, so we thought things will develop as expected, so that his retrograde amnesia would kind of dissipate over time, and his anterograde amnesia will be more persistent. In fact, over time, the opposite happened. And furthermore, if anything, his retrograde amnesia was even more severe than in his anterograde amnesia. He didn't know. You would ask him, who wrote King Lear? He wouldn't know. What is the capital of France? He wouldn't know. He was, he grew up in an upper middle class professional family, went to a good school. Children like that usually learn this stuff way before the age of 17, okay? He didn't have this knowledge. What was also peculiar, that his knowledge was impaired both for verbal memory and for visual memory. You would show him some kind of various, emblems, various symbols like Rolls-Royce symbol or swastika or Olympic rings. He had no kind of a non-verbal kind of a symbol. He had no idea what they were, okay? So we decided that lest people refuse to believe us, we better document it, okay? So we documented it. I don't want to go through the details of this slide, but it captures this pattern that I just described. Anterograde amnesia improved very drastically. Retrograde amnesia remained very severe, did not budge. And we sent this article to Science magazine, okay? And of course, Science and Nature are the two most august journals in the world. And today, virtually every issue of Nature or of Science has something about the brain. In those days, that was not the case. That was, when was it published? In 1981. In those days, basically, neuroscience was not even regarded as a legitimate science, okay? It was all about physics and chemistry, and you did not see neuroscience papers in these two august journals. We had the chutzpah to send it to kind of the goal to send it to Science. And they accepted it. It was published there, but they had one concern, and it was my concern also. I remember one of the reviewers said, well, you are claiming you described this global memory impairment, but if you look at the lesion and the lesion which accounted for it, and that's also an interesting story, but yet a separate story, maybe for another day. It was a mesencephalic lesion between the pontomesencephalic junction on one end and the ventral tegmental area in the other end, and basically it obliterated the ventral tegmental area, which sent these dopaminergic, but not only dopaminergic, mostly dopaminergic, but not exclusively dopaminergic projections to the prefrontal cortex and to the hippocampi. It's a very important area. And we were actually among the first. Now it's all the rage. Everybody studied this ventral tegmental area. It was basically completely obscure when we reported this. So we're among the first ones to study it. So, but this lesion is basically lateralized. It's lateralized basically to the left hemisphere. Here, left is left and right is right, okay? And one of the reviewers basically almost basically nixed our paper, saying it's inconceivable that a lateralized lesion would result in such a global deficit. And I had to engage in all kinds of mental gymnastics to explain it, to that, at the level, VTA level, it got the midline, blah, blah, blah. But I knew that basically he was right, that there was a problem there. But the paper was published, very nice. But that, at the same time, it basically suggested the damage to the left hemisphere may wipe out access to information, long-term memory store, which is both verbal and non-verbal. And then we began to examine it more systematically. And basically, in a series of studies, I don't want to go into too much detail. These are studies, this is a doctoral dissertation by my former student, Bill Barr, who is now William Barr, who is now head of neuropsychology at the New York University School of Medicine. He studied patients with temporal lobectomies, okay? And basically demonstrated, looked at retrograde amnesia in temporal lobectomies, and basically demonstrated that right temporal lobectomy, and of course we all know that hippocampi are very important for memory. Basically what he demonstrated, that right temporal lobectomy, which involved the hippocampi, did not particularly interfere with very remote memory, did not cause retrograde amnesia. Left temporal lobectomy did, and it was true both for verbal and non-verbal information using various techniques. Again, normal subjects, right temporal lobectomy, left temporal lobectomy. Take my word for it. These are, I don't want to spend time talking about the details of these specific paradigms, but this is the kind of a task that in order to do well on this task, you need to have access both to verbal and non-verbal memory. But according kind of the old precept, damage to the right hemisphere, left hippocampus would interfere with verbal memory. Damage to the right hippocampus would interfere with non-verbal visual-spatial memory. So damage to either one would interfere with the performance on this task, but it didn't happen, okay? Damage to the left hippocampus interfered both with verbal and non-verbal memory. Damage to the right hippocampus was neither. And so we had a whole bunch of these tasks, all of which demonstrated that, okay? So now we had very systematic evidence that damage, that somehow long-term store somehow depended on the left hemisphere, whether it was verbal or non-verbal, which of course goes contrary to this old kind of a classic old school notion about this double dissociation, verbal memory as an extension of verbal processing linked to the left hemisphere, non-verbal visual-spatial to the right hemisphere. That's not what happened, okay? And it all started with this mysterious case where the lesion was lateralized, but the deficit was global, okay? That's what really kind of planted this question in our heads and then triggered this kind of more systematic line of research. And if you look at agnosias, basically the same thing comes through. Agnosias are, psychologists among you are familiar with this term. Agnosia is a very generic, very general term for deficits of higher order perception in the absence of kind of a more basic psychophysical impairment, right? That's what agnosias are. Very broadly speaking, we distinguish between associative agnosia and apperceptive agnosias. Associative agnosia is basically an inability an ability to identify an object in terms of its kind of a basic sensory attributes. A patient with associative agnosia will tell you it's yellow, it's long, it has a sharp end, and it's a dull end. Basically, a pretty good account of kind of a sensory attribute. And then he will say it's a cigar or it's a knife. So he will be unable to identify it as a member of a generic perceptual category. Okay, so this is one kind of agnosias. In other words, agnosias whose existence is interpreted by most clinicians, by most scientists, as a kind of a breakdown of the generic representations, long-term representations, which allow you to identify, to encounter a technically de novo object and immediately recognize it as a member of a familiar category. Okay, so these are associative agnosias, as opposed to apperceptive agnosias, agnosias which do not, which basically entail the impaired ability to recognize an object as its own self under various conditions of observation. If I show you this thing, you will, you'll recognize it. If I show you this thing like that, you will recognize it as the same object, even though its retinal impression has changed. All right. So these are different types of agnosias. So, and the pure agnosias are very rare. So whenever one is encountered in a patient, it merits a journal article. So with this Bill Barr, he was no longer a student of mine, a colleague of mine. We conducted a literature review trying to fish out all these cases of isolated and isolated perceptive agnosia and associative agnosias. And according to the textbook, it will tell you that these two forms of agnosias are caused by bilateral damage to the posterior aspects of the hemispheres. That is true. But what we found out, and basically it was known, sort of known before also already, that whereas a bilateral lesion is certainly a sufficient condition for these agnosias to ensue, it's not a necessary one, that they may be caused by a lateralized cognition also. And when that happens, there is a double dissociation. So the damage to the left hemisphere, occipital temporal regions in the left hemisphere, will result in associative agnosia. In other words, that form of agnosia, which is a product of this breakdown access to previously formed generic kind of categories, and the damage to the right hemisphere will result in apperceptive agnosias. So again, the fact that associative agnosias, that this access to the generic categories may be impaired following damage to the left hemisphere, sort of suggests that these categories are somehow mediated predominantly by the left hemisphere. So it appears, just to summarize all these various strands of evidence, kind of a literature review of agnosias, and our systematic studies of patients with temporal lobectomies. And it all began with this unusual case, this young man with a horse riding accident. Basically, it all led to the conclusion that damage to the left hemisphere interferes both with verbal and non-verbal old established knowledge. Okay, and I talk about it in this old book of mine, The Executive Brain, which was published in 2001, I think, and it was translated into Russian under this title, Upravляющий мозг. And I was told, I was very gratified to, and I'm saying it for a reason, I was very gratified to find out that this little book is used practically as a textbook at University of Moscow. and possibly at other universities. And Novosibirsk, what Professor Zelman put me in touch with these people of Novosibirskом, как называется, Novosibirский государственный университет, yes. And the first thing she did, this professor, she propped up this old book of mine, so it's used there also. So I'm very gratified to know that. But there is a new edition of this book titled The New Executive Brain, which is much expanded. It's much thicker and much expanded. And to the extent that this book was deemed useful, this newer book would be so much more useful. So it's too bad that it's a shame that it hasn't been translated into Russian. So we need to somehow contrive to translate it into Russian and also make it available to students. So, and it's also this notion linking established knowledge to the left hemisphere is described in this book, Управляющий мозг, and also in this book, The Wisdom Paradox, which has also been translated into Russian. Paradox мудрости, yes? So just to summarize what we said so far, and then Matvey will tell me when to quit, because this is a modular kind of a talk. I can stop at various points, and you wouldn't even know that I had more to say. So the neuroanatomy of cortical representation is not static. As information becomes more familiar, the underlying cortical representations change. And this has profound implications for how we process functional neuroimaging data. I assume that at least some of you do this work, or at least read about this work. So basically what we do, subject is in a scanner, and you engage the subject in some task, and then you average, basically average across all data points in the course of this experimental sequence. And that's what you do. And of course, that's understandable. You have to maximize the number of your data points. The signal is very weak. The signal to noise ratio is very faint. So of course, it behooves us to maximize this ratio by increasing the number of data points to the extent possible. However, this enterprise is justified only to the extent to which you can be assured that these various data points come from the same population. If they don't, then what you are getting is garbage in, garbage out. You see what I'm saying? It's like, imagine you have Johnny, who was at the age of 50, at the age of 10, he weighed, well, let's say, how much does a little boy weigh? 50 pounds, okay? Then in his adulthood, he was so fond of beer and sausages that he ballooned to 200 pounds, okay? And then in his dotage, he again shrunk back to 50 pounds. So what do? You add 50 to 150, you get 300, you divide it by 3, you have 100 pounds. And you say, Johnny weighs 100 pounds. Sounds plausible on the surface, but totally meaningless data. You know what I'm saying? Because the Johnny's weight changed in the course of his life, and averaging across his lifetime, and then coming up with a single static constant is basically, it's it's meaningless. These are meaningless, misleading data. So I am afraid that this is something similar happens with the way we process the functional neuroimaging data, as long as we average across the whole sequence. Because, this whole idea to average across the whole sequence, as it's very seductive, it's very attractive, because you want to increase the number of data points, but it's predicated on the assumption that you are pulling these data from the same neural structures. But if this is not true, and what is true instead, that the subject, in the course of doing a cognitive task, the underlying neural structures change, then you cannot do that. You need to do something entirely different. Instead of dealing with some spatial constants, you need to develop a totally new methodology of what I call spatio-temporal vectors. You need to segment this sequence and then average within each segment, and then create vectors out of these segments. Okay, very different way of processing data processing. And whenever I talk to colleagues about it, they resist the idea because you will indeed sacrifice kind of the robustness of your data, because for each of these segments you will have fewer data points. But I truly believe that this is the only way to kind of legitimately process data and new functional neuroimaging data, and without it you'll get the proverbial garbage in, garbage out. So it's necessary to develop a new dynamic functional neuroimaging methodology, segmenting the experimental sequence instead of averaging across all of it. Okay. If Matvey gives us more time, we'll talk about evolution. Does Matvey give us more time?
Yeah, I have no right to stop your lecture, but maybe
No,
we'll go through a couple of questions.
You have every right to disallow me to abuse the patience of my audience. If you think that it's time, you have your rules. If it's time to quit, then we quit. As I said,
No,
it's a
let's
different
proceed.
module. It's a different module. I can stop here. And you would not have even known that I had more to say, because it's a finished kind of a clause, and now I want to talk about something different.
Okay.
Yes or no? Quick.
Yeah, we still have 10 minutes, so
Okay.
if it's okay.
Then let's talk about evolution. This whole enterprise, this whole kind of embrace of this formulation of this idea and embrace of this idea was really driven by evolutionary considerations. I was adamant about somehow hitting upon some principle which would be meaningful across throughout evolution, and not just limited to humans, because only humans speak language. Okay, these were the origin of this whole novelty-routinization idea. So, so what I want to do now is to see if there are some data about hemispheric specialization in other species. Okay, and indeed, what you will see over the next few slides, that novelty seeking is linked to the right hemisphere across multiple species, and kind of a practice of cognitive routines is linked to the left hemisphere across multiple species. So, and again, I talk about it in this creativity book of mine. Now, this notion, the first systematic review of this subject was conducted by McNeilage, Rogers, and Vladimir Zelman, Origins of the Left and Right Brain. It was published in Scientific American a number of years ago. And, to their credit, they give me credit for kind of a starting this whole kind of a line of reasoning. But they were the first ones who conducted a systematic review of that subject. And then, that was a long time ago, so I sort of conducted my own review for this book, Creativity. Indeed, it has been demonstrated in primates, in baboons, that basically what the Fagon and Vauclair did, they put a baboon in an experiment which was basically designed by humans, kind of a tachystoscopic setup, where the baboon was taught to kind of put its chin on the chin rest and fixate on the fixation point, kind of a low-tech technology which we used to use before the advent of neuroimaging. I used to do this kind of a very low-tech experiment at Rockefeller University in New York when I first came to New York, right? So, and then this monkey, and baboons are not apes, they're monkeys, they're just big monkeys, was taught to fixate on this central fixation point, and stimuli were flashed at very brief intervals, maybe 50, 60 milliseconds to the left or to the right, and there was some task to recognize them, and the comparisons were made which presentations were more effective. And it turned out that, again, in the course of this experiment, stimuli delivered to the left hemifield, namely ostensibly to the right hemisphere, were processed more accurately, and by this monkey and by this baboon. And then the monkey learned there was this transition from the kind of a right hemispheric, left visual hemifield, right hemispheric superiority to the left hemisphere, right hemispheric superiority. Basically, the pattern was very similar to what was demonstrated by similar studies in humans, in the 70s, 80s, people were infatuated with these paradigms. Um, yeah, so geometric shape discrimination, that was the task. In baboons exhibited this kind of a transition from the early right hemispheric superiority to subsequent left hemispheric superiority. In dolphins, exploratory behavior is guided by the left eye. And of course, dolphins are mammals and their pathways are also crossed. So left hand, and their eyes are much more lateralized on the kind of a lateral aspect of their muzzle, of their snout. Whereas numerosity, which was experience-based, was mediated by the right eye. Again, overlearned skills were kind of depended more on the right eye, which connected to the left hemisphere. Pathways are crossed, and exploratory behavior, where they were exploring, they were mostly relying on the left eye. And even in the lowly rats, in the rodents or in the mice, spatial memory and hippocampi revealed a similar relationship. That as a rodent learned developed some spatial memories, it relied increasingly on the left hippocampus. Okay, so a wide range of mammalian species exhibit fundamentally the same principle: exploratory behavior and kind of a novelty exploration linked to the right hemisphere, processing with reliance on established routines, left hemisphere, just like us, right? And also in avians, and of course in birds, avian brain is very different. It has very little, if any, cortical development. Okay, it's basically it's a non-cortical brain. It's a bunch of nuclei, large thalamus, large striatum. It's basically it's a collection of nuclei. Okay, so a very different kind of a neural organization. Yet this division of labor between the two hemispheres appears to be very similar. So when a bird is exhibited to a novel song, a song that it has not heard, zebra finch, for some reason, this bird is beloved by experimenters. It's used in avian experiments, like rodents are used in human in mammalian experiments. So when a zebra finch hears a novel song, a song that it had not heard before, the right hemisphere becomes activated. When it hears a familiar song, the left hemisphere becomes activated. Sorry for the typo, okay? The more rewarding the stimuli are, the more they engage the left hemisphere. So again, there is this the division of labor, which is consistent with this novelty-routinization hypothesis, and even in invertebrates, okay? Like in honeybees, memory for odors, for odors, so that if you expose this creature, this honeybee, to some new odor, and they teach it to recognize it, then in short-term recall will rely on the right antenna, and in these creatures, their rudimentary nervous system is uncrossed, okay? And the long-term recall on the left antenna. Again, this transition from kind of a immediately after the exposure, reliance on the right antenna and then left antenna. So this appears to be a totally, and they just for the hell of it, I included this picture in my book. It appears that these very, very different creatures, primates and dolphins and birds and insects, they all exhibit this division of labor, so that the right hemisphere, or whatever passes for the right hemisphere in a creature like a honeybee, is somehow engaged in dealing with recently presented novel information, and the left counterpart overlearned information. Okay, so it truly appears to be a universal principle. So is it a conservation of an early principle or a convergent evolution? I don't know. Okay, it's an interesting question. I don't have an answer. It may be a conservation of an evolutionary principle, or it may be convergent evolution because it is so adaptive to separate as you know, computational neuroscientists, neural net modelers have established on their kind of a models that it's very adaptive to separate these functions. But either way, this novelty familiarity division of labor appears to be universally adaptive within the central nervous system, however its organization is, okay? And again, let me just say where we started, that we are so used to this narrative treating every aspect of hemispheric specialization as secondary to this distinction between language, secondary to this link between language and the left hemisphere. But you know, basically, I believe, and based on what I try to present here today, language lateralization itself may be a secondary to a more fundamental principle linking cognitive routines and long-term knowledge to the left hemisphere. And of course, a language is a very complex system of cognitive routines, okay? And distinction between language and nonverbal functions is merely a special case of this more fundamental distinction between the two hemispheres, which is meaningful across species. And as I said, it's kind of, I'm talking about, it sounds nostalgic because this whole exploration started when I was probably 19 or thereabout as a sophomore or third year student at University of Moscow. And it sort of continued as an enduring theme up to this point through my career. Now, if Matvey gives us more time, we can talk about the underlying neural architectures. If it's time to quit, we will quit. As I said, this lecture was designed intentionally in a very modular fashion so that I could stop here. You would not even know that I had something else to say. So if you feel that we should stop here, we'll stop here.
I propose to answer several questions we have in Q&A, and then we decide, okay?
Then you decide.
Yeah, together with attendees, because, you know, we are quite democratic, so we'll vote.
Yeah, well, I like to tease young people, yeah.
So let's go briefly through the questions we have in Q&A. And also I would like to mention that anyone can raise a hand and we'll give a mic to this person. You can also put...
okay, so I need to click. What do I click? Q&A? There are 9 questions.
Let's pick several of them.
Hang on. Okay, now I see. AC, during right to left hemisphere connected. Yeah, that was the question which we already saw. And as I said, I don't have an answer to this question, but it's a very good question. And maybe one should design a study. Thank you for the idea, AC. Okay. Dear Professor, thank you so much for the opportunity, partners. Sometimes I face with unusual and novel side effects of L-DOPA in my patients with Parkinson's disease. Male patients with gambling, this is a very interesting subject, and in fact, one of these modules to which we definitely won't have the time to get was about hemiparkinsonian syndromes. But let me read this. Sometimes I face with unusual and novel side effects of L-DOPA in my patients with Parkinson's disease up to severe. Male patient with gambling addiction on L-DOPA only or female with hypersexuality. But you told us the dopamine involves in the routine and familial processes. But there is no history. Does it mean, okay, does it mean that there was a latent predisposition to this addiction? Thank you in advance. This is a very interesting phenomenon. It's known that not all, and probably not even most, but there is a subset of Parkinson's patients who, when they are put on dopamine agonists, they develop addictions. They gamble, become gamblers, or become sex addicts. These are, or they develop this phenomenon called punding. In a nutshell, let me give you the kind of a short version, and then I can enlarge. In a nutshell, what these addictions are, as far as I'm concerned, are perseverations. They develop, and I think that this is really captured in this phenomenon of punding. What is punding? A patient with Parkinson's disease will take a gadget, whatever, like this pen. He will disassemble it, assemble it, disassemble it, assemble it, disassemble it, assemble it. Go through these perseverative routines over and over again. This is perseveration. I believe addiction is commonly understood, at least in the United States, as impairment of impulse control. I think that it's only half the story. The other half of the story, and maybe even a larger part of the story, is perseveration. These addictive behaviors are perseveration, stereotypic behaviors. Now, where do they come from? I would imagine that there are some predispositions. How will, so that if somebody develops hypersexual, well, sex is something that we all engage in, okay? So saying that somebody was not predisposed towards sex before is probably incorrect. I mean, the person was not a sex addict, but a person engaged in sex, all right? And gambling is probably also some kind of a manifestation. Yes. Can we go back to this question? This question disappeared.
It's an answered now.
Huh?
So if you click answered,
Well,
and then you can see it again on the bottom.
okay, right. So I think that these I think that these addictive behaviors are a form of perseveration, and the form and perseveration is stereotypic behavior. We know that in patients, and again, that's part of my one of the kind of forthcoming modules to which we will not have the time to get to. That at least in males, damage to the right frontal lobe, prefrontal cortex, will trigger exploratory behaviors. Damage to the left prefrontal cortex in males will trigger perseveration. So I think that these addictive behaviors are perseverations. Matvey, can he or she hear me, the author of this question?
I guess so, yes.
Then I'll ask you a question. Did you perchance notice whether these addictions, what you call addictions, are more common in left or right hemiparkinsonian patients? Of course, we know that most cases of Parkinson's disease are in fact hemiparkinsonian cases. The tremors are almost never of equal severity in the two hands, okay? They're usually more severe in one than on the other, hence hemiparkinsonian syndromes. Usually more severe in the dominant hand, but not always. So my question to you is, have you noticed, or if you haven't, will you from now on start paying attention and then letting me know sometime in the future, Matvey will tell you how to get in touch with me, whether this propensity toward addictions are more common in patients with left hemiparkinsonian kind of manifestations, in whom, one would presume that this atrophy of substantia nigra is more pronounced in the right hemisphere, and the depletion of this nigrostriatal pathway is more pronounced in the right hemisphere, or in the patients in the or in the right hemiparkinsonian patients, in whom the opposite would be kind of a positive, that this depletion of nigrostriatal dopamine would be more pronounced in the left hemisphere. I would be very interested to know. All right.
Okay,
Next
this was a
question.
question to Mark Kachanovsky. So next question, what set the functional difference between right and left hemispheres in the embryonal development?
Interesting question. I don't know. I don't know.
Okay.
Again, is a worthy question.
Mhm.
Grigory Petrov, what is the approximate time of number of repetitions for novelty to become? That depends entirely on the nature of the task. This depends entirely on the nature of the tasks, which is why I said that you probably have these kind of curves, right to left shift curves, different time scales for different tasks. It's not a single kind of a curve. Okay.
So how can one use the information on how brain processes information, novel routine, in order to have some practical beneficial outcomes, such as studying strategies, cognitive enhancement, devices, etc.?
Well, we need to think about it together. Some people like Alan Snyder feels that that to the extent that you need to enhance your ability in certain situations to deal with cognitive novelty, you may either suppress the left hemisphere or kind of a less radically maybe enhance the right hemisphere. I think that such knowledge of cognitive styles is very important in many ways, but one obvious way is for the composition of optimal teams. when you need to have people of various kinds with various talents? It's an interesting question. I have some ideas which we can discuss. It will take a long time, all right? I talk about it to some extent in my book, This Creativity, which has been translated into Russian. Yeah.
Okay, then maybe you need to visit one of our neurobars to discuss this point.
Maybe.
Mhm.
What do you serve in your neurobars?
Everyone serves for himself or herself.
I see.
Mm.
Дорогой Элкхонон Абрамович, большое спасибо, лекции. Ваши книги я рекомендую своим студентам. Ну, спасибо, что вы рекомендуете мои книги своим студентам. Нужно как-то это организовать, чтобы New Executive Brain перевели. Вот мою книгу по креативности перевел Эксмо. Вот поговорите с Эксмо. Это, сколько я понимаю, такое важное издательство в России. Пусть они мою новую книгу тоже переведут. Это для ваших студентов было бы очень... Это было бы лучше, чем вот этот управляющий мозг, потому что это куда более новая книга, и она более расширенная. Вихай Кумар.
This was just some comment in Russian for those who doesn't understand.
Student, student or just stud. People are currently have so much novelty around them thanks to social. In your opinion, how can this change brain principles or of perception affect hemispheric organ, and what outcomes can be prognosticated? This is a very profound question. We have no idea, not enough work has been done to study the effects of social media, of these various virtual methods of communication like on the brain. This is a very important question. The literature is very kind of accumulating, but very slowly. So I cannot answer this question, but I completely agree with you that it's a profoundly important question. Anonymous attendee, planaria flatworms have left and right cerebral ganglia, and did the simplest living animal having a body plan or bilateral asymmetry, encephalon and exhibit long-term. Are you aware of it? I am not, but this would be obviously kind of the next natural question to ask. If you do work with planaria flatworms, maybe that could be an interesting research topic to see whether there is, when we're saying that it's bilaterally symmetric. Yes, it's in a very macroscopic sense, humans are also bilaterally symmetric. We have the two hemispheres are more similar than they are alike. But then the next question is whether there are some subtle asymmetries. So planaria flatworms were not part of my review. I just haven't come upon this literature, but maybe it exists, maybe it doesn't exist. If it doesn't exist, I think that it would be a very reasonable question to ask. Artyom Dyerentsy, speaking of the damage to the frontal lobes, hang on, it's a different one. How do you think neural novelty familiarity dichotomy would turn out in a person subjected to corpus callosotomy? Thank you for your answer in advance. Loved your book and your lecture. Okay. I'll give you a short answer and a long answer. The short answer, I don't know in an empirical sense. I have not conducted these experiments, nor to my knowledge did Michael Gazzaniga and these people who in his group, Zaidel and all those, Iran Zaidel and others, who studied callosotomized patients frame the question in this fashion. They really operated with this traditional language visual spatial paradigm. I personally, but what I can say that the man who basically supplied all these patients, Joseph Bogan, of course, he's slobbering. Michael Gazzaniga and Diran Zaidel and Jerry Levy, and Jerry, well, it all started with Roger Sperry, of course. It was the group who studied these callosotomized patients and became famous because of that. But the man who, but then there was this man named Joseph Bogan, who was the neurosurgeon in Los Angeles, who conducted these surgeries. Professor Zeldin, did you know him by any chance? Joseph Bogan?
Yes, I know him very well.
Well, he
Actually,
died a
he
number of
worked
years
in
ago.
our department of neurosurgery. The chairman was Dr. Martin Weiss.
Yeah, and
And,
he was very, he
also, he
became,
was in the Huntington Memorial Hospital and the county hospital.
right. So he was the surgeon who supplied all these colostotomies. He became very interested in my novelty-routinization theory. He truly embraced it. We became friends on that basis. He was much older. He died probably at least 15 years ago, I would say. But he was very interested in that. My speculation would be that, that if you subject, suppose you take a colostotomized patient, put him or her through some kind of a formal experiment when the patient is supposed to acquire some new skills. Okay, I would say that if the stimulus is presented to the right hemisphere, the task is presented to the right, there will be learning up to a point, and it will plateau, it would asymptote very early. If the learning is mediated only by the left hemisphere, there will be no learning. Okay, there will be just flat curve from the get-go. In terms of real life activities, the interesting thing with these colostotomized patients, and I've seen my share of patients with callosal agenesis, with corpus callosum. I didn't didn't study them as a researcher, but I just saw them as a clinician because I've been seeing patients of all kinds for many years. In terms of real life activities, there don't seem to be any major kind of a deficiencies. But you have to keep in mind that just colostomy alone does not preclude communication between the hemispheres. Commissures remain, anterior commissure and posterior commissure, and some communication may also take place on the striatal and thalamic level, even though there it's very limited because these thalamic nuclei are notorious for not having extensive connections. So that's my answer. We can talk more about it again, but maybe some other time, all right? So I can answer some other questions as well.
Yeah, we have two more questions in the Q&A.
Now, I have general question. Do you think that animal can also think like humans or they don't? Well, I had read somewhere that in order to think it's necessary to have language, and animals don't have language. Listen, thinking or this is not a binary distinction. It's not that either you think or you don't think. There are degrees and qualitative differences and quantitative differences. I've never studied. Well, that's actually not true. At University of Moscow, I did some work with cats, but that was fleeting kind of activity. So the bulk of my research career has been with humans. But I am a lifelong dog owner. I have this big animal sitting right next to me in my office, Brutus. Occasionally he slurps or snores. You may have heard him in the background, for which I apologize. He's an English Mastiff. He weighs 90 kilos, not 90 pounds, 90 kilos. And I've had dogs my whole life. They can think, I'm telling you. They are sentient creatures. They have emotions, they have thoughts. Are they like humans? No. Do I understand what's happening in their heads? No. Do they understand what's happening in my head? No. But as somebody who has interacted with dogs my whole, well, or at least much of my life, they are sentient creatures. Okay. So just as a human being who has had an animal around me much of my life, I believe they can think. All right. Nikita Glukhov. Speaking of the damages to the frontal lobes that you mentioned earlier, at this point of time, do we understand the mechanism that produce such interesting cases that a person can acquire more skill, become more intelligent in an area than he or she not good before, after the damage? Well, again, understand, don't understand, we shouldn't phrase it in binary terms. There are degrees of understanding. Like the example that I brought up with frontotemporal dementia, some people think that, well, first of all, I don't think that somebody would develop an artistic talent without any prior interest in the art. So it's not as if some kind of a new skill appeared like Deus Ex Machina out of total nowhere, but some kind of an enhancement, maybe a result of pathology. Okay, no open questions, only hosts and panelists can see the questions.
Yeah, which means that this was the last question. And actually, we're out of time. So I would propose to close at this point. And this will be another motivation for you to come to Russia and to give a lecture in person. What do you think?
I think that when the pandemic will subside, maybe I will. Yeah.
Okay. Yeah, another reason is that now it's almost 7 p.m. in Moscow, which means that Novosibirsk is already sleeping. That's why we don't have all of the participants right now. And Moscow people, they probably would like to go to think of what they just heard to some bar or I don't know, whatever place. So if you don't mind, let's stop at this point. I think this was very interesting 3 sections.
Okay, well, so anonymous attendee, will you have a record? I will definitely revisit it.
Oh yeah, we will have a record, but the record will be available, I don't know, in 1 or 1.5 months. If we have anyone who has a question and want to raise a hand and ask orally, we still can do this. Let's do it, like, still have a couple of minutes. And if not, then yeah, I would like to ask if maybe Mikhail or Professor Zelman, you have any questions to Dr. Goldberg?
I would just thank you very much, Dr. Goldberg, and to you, Matvey, for organizing all these things. And I believe it's very, very informational, and I hope that we will continue this tradition to unite the world of knowledge, science without borders.
Yeah, sure. Thanks.
And I really enjoyed the talk. It was great, and I had some idea. I even had some ideas of experiments of
Excellent.
building better brain-computer interfaces based on these ideas.
Well, maybe at some point we can talk about one of my kind of a hidden modules, which we didn't have time to talk to, about the underlying structural difference between the
Yeah,
and
this would
possible
be really
implications
interesting.
for neural nets and kind of AI architectures.
Yeah, great.
Yeah.
I see one raised hand and one more question in Q&A. Let's answer them and then finish. So first of all,
stress affect the different hemispheres? I don't know. A very good question. I don't want to ad lib here. I don't know.
Okay, and one raised hand, so I will allow you now to talk, but please be polite. Do not say bad words. You're free now. Yeah, to unmute.
Yeah, can you hear me?
Yes, we can hear you.
Yeah. So thanks for the talk. So my question is that
can you just increase the volume a little louder?
yes, sure. Is it audible now? Yeah. So
Yes.
my question is So my question is with regard to Ramanujan's claims that he used to see formula from deity called Namagiri. So what's your opinion on that? Like he come up with the.
Can you do me a favor and type this question? It's not a very good reception.
Okay, sure.
Matvey, can you give her an ability to type the question?
Yeah, sure. It's open. Sorry, we cannot hear you really well, so it's some background noise. Maybe you'll type this question. Okay, let's give 1 or 2 minutes to do this. Let me, in the meanwhile, introduce some. So thank all the participants of today and other seminars. So this is the last seminar of CNBR Open project for this year. And we'll continue in the next year. And moreover, I think we'll make some New Year's lottery for the participants. So everyone who is subscribed to our mailing list will participate, and we will also definitely. So some of your books, Dr. Goldberg will be the presents for people.
I'd be delighted. I feel honored. Yeah.
Okay, no, we don't have the questions so far. Let's stop at this point. Let me
Yeah, I
again.
apologize to the person who had the last question. I just couldn't hear what you were saying.
No, now we have it. So it's in chat. What is your opinion on Ramanujan's claim that he used to
Ramachandran.
sim?
Hang on, let me open it. Yeah.
So it's in the chat.
Yeah, I'm trying to click on it and it's not opening. It's not opening.
Could you please resend it to Q&A? Just copy paste.
Professor Zelman, so you knew Bogan. You knew Joseph Bogan.
Yes, I know him very well.
Well, we became very good friends. Okay, what is your opinion, is it Ramachandran? Ramanujan, I don't know the spelling, claimed that he used to see mathematical this great mathematician with the help of goddess. I have no opinion. I'm not a great mathematician, so he saw a mathematical formula with the help of a goddess. Well, if he did, so much more power to him. What can I say? That's why he was a great mathematician and I am not.
All right.
But I'm very interested in cross-cultural cognitive neuroscience. So I'm very interested in kind of such beliefs and the brain mechanisms. I don't have to share these beliefs to be interested in them. We'll conduct now research in Indonesia, for instance, motivated by this kind of a cross-cultural interest. So, I find it interesting.
Okay, so at this time we don't have any question in Q&A and no raised hand. So my proposal is to close now. Thanks again for you, Professor Goldberg. Have a nice day. Sorry, it's just the beginning of the day on your side. I would also like to thank Professor Zelman for for joining us in such an early hour. Really appreciate this, and thanks for what you do for the seminar and your support. And thank you, Professor Lebedev, for also supporting the seminar and joining today. Yeah, see you next time in the new year. Goodbye to everyone.
Bye. Happy New Year to everybody, and let's hope that the next year will be happier than this one with this pandemic and all this nonsense. Yeah.
Okay.
So bye-bye.